Hello, in this video I want to talk about the resting membrane potential of a neuron. So the resting potential of a neuron is about -70 millivolts. And there are a couple different things that have a major influence on resting membrane potential. Number one are proteins. Proteins are negative and they're found inside of the cell, not out. Number two is the sodium-potassium exchange pumps. There is a third thing that contributes to resting membrane potential, and these are called ion leak channels, but I won't be talking about these in this video. So let's first look at proteins and how they influence resting membrane potential. As I mentioned, proteins are negative, which means they're anions, and about 94% of all proteins are found inside of cells and not out, which means that we have these large, bulky, negative things inside of our cells. This makes the inside of a cell much more negative than out. The resting membrane potential is around -70 millivolts. The negative means that the inside is negative compared to the out, and you can see how the negative proteins contribute to this. So in this picture here, we can see a cell membrane, so that there's a phospholipid bilayer with proteins embedded in it. We're going to call the upper part extracellular and the lower part intracellular. Notice that we have a voltmeter measuring the difference in the voltage between the inside and the outside. And the difference is being measured at -70 millivolts. So we were just talking about proteins, right? So if you look at the bottom of the screen, we see we have these large pink blobular things, and I have them labeled as a protein. And then they have little negative signs on them. Proteins are negative and they're found inside of the cell. And that's one of the reasons why we have this resting potential that is negative, because of these negative proteins. When we talk about a membrane potential, by the way, we always talk about what the inside looks like compared to the out. The inside looks negative compared to the out, in part because of these proteins. Now let's take a look at how the sodium-potassium exchange pumps can influence resting membrane potential. Sodium-potassium exchange pumps are proteins that actively transport sodium ions out of the cell and actively transport potassium ions into the cell. The sodium and the potassium don't move at a 1-to-1 ratio. 3 sodiums are pumped out for every 2 potassiums that come in. These pumps require energy because we're actively transporting sodium and potassium against their concentration gradients. More than 50% of your food energy goes into running these pumps, so they are really important for your neurons and muscles to function properly. So let's take a look at why the sodium-potassium exchange pumps contribute to the -70 resting membrane potential. If we're pumping 3 sodiums outside for every 2 potassiums that come inside, we're going to end up with more positiveness on the outside because sodiums have a positive charge and potassiums have a positive charge. So you're putting 3 pluses out for every 2 pluses that come in. So more pluses outside than in, hence the inside will have to be more negative compared to the out. This is also important, the sodium-potassium exchange pump is also important because it establishes a concentration gradient for sodium to enter neurons and for potassium ions to leave neurons, which we will talk about more when we get to graded potentials and action potentials. Well, let's take a look at a picture that describes the sodium-potassium exchange pump. So you can see we have a sodium-potassium exchange pump embedded in the cell membrane of our neuron. And it's going to pump 3 sodium ions out. The sodium ions are shown as green circles with a plus. For every 2 potassium ions that come in, the potassium ions are shown as a red plus. And notice that we have a higher concentration of sodium outside and a higher concentration of potassium inside. So the sodium-potassium exchange pump is helping to establish this negative resting membrane potential by putting more positiveness on the outside than the in.